Buddy, thank you very much, Dr. Ashutosh, and the entire Cardiabcon team for giving me this opportunity to talk to you on some of the most important things that I thought that are probably not addressed properly during this COVID time. Is the use of monoclonal antibodies. Um, They have come, they have tried to establish themselves. There were some challenges which were. So let's try and explore, how and what this COVID has taught us as far as the monoclonal antibodies and its use during these difficult times. So what I'll try and do in this 15-20 minutes of my talk is this, that I'll just give you an overview of our immune system, and not only that, where you place this monoclonal antibodies, why at all they have to be used, and what was the concept that we thought that monoclonal antibodies would be useful, especially managing this demon of COVID-19. Now, friends, we all know that we are at war, always invisible war. Since the time we take out first, we are at war. We are at war with multiple things, and especially these microbes. But then we keep on winning this war. We successfully defend them thanks to our immune system, which works in tandem, in synchrony, like a symphonized orchestra. And it comprises of huge system, comprises of various cells of immunity, comprises of various proteins of immunity, comprises of various chemicals of signaling and communication. And they all do what? They're in an orchestra of defense, which works in tandem in beautiful symphony. A very interesting fun fact I came to know when I thought that let me know how much is the daily blood WBC production in the blood. We all know that WBCs are the pivotal cells for our immunity, comprising of neutrophils, eosinophils, basophils, monocytes, which are also having natural killer cells, dendritic cells, T lymphocytes, B lymphocytes, and so many other things. But when I looked at the daily WBC production, blood WBC production, I was mesmerized. Whooping 370 billion cells in 24 hours. You know how much is the global population of human beings? 7.8 billion. Imagine, nearly 7 to 8 times of world population WBC production happens in our bone marrow. And it happens through a pluripotent hemopiotic stem cell, giving us different lineages of leukocytes, lymphocytes, and platelets. And I used to always wonder that how they work tandemly as a synchronized orchestra. And when I started exploring, I realized that they communicate with each other by a sort of a chemical signaling which happens through what we know as cytokines and chemokines. Now, cytokines are the intricate chemical signaling system, which are of 4 different types, like interleukins, tumor necrotic factors, interferons, and the colony site growth stimulating factors. Remember that primarily these all cytokines, they work to either bring out the change in the cells of the immune defenses, like they bring out multiplication, activation, maybe transport, maybe apoptosis. So they bring out biological change in the cells of the defense system, especially in the lymphocytes. And chemokines, they work as Google Maps. Chemokines direct the cells of immunity towards the site of the insult, and they work in tandem. So if you really look at the COVID situation, you will see that the moment the COVID virus enters our respiratory system, they usually target the nematocytes, especially type 2 nematocytes, which are also mucus producing cells, and they start multiplying there. The moment they attack these pneumocytes, they are sensed by something called as toll-like receptors, and the message is sent to the dendritic cells, and they trigger the genesis of CD4 T cells response. And then CD4 will call for T helper lymphocytes. Now, T helper lymphocytes on one arm will call for macrophages, but also will stimulate the virus-related antibodies, which are virus-specific from their memory cells. So they'll call for the memory T cells in action, and also will trigger and stimulate the production of CD8 T lymphocytes, which are also called as natural killer cells. They'll go there and do what? They'll engulf the insult of by the COVID-19 virus, and the process is over. The systematic defense happens. But in severe COVID, what happens is this, that there is an excessive production of pro-inflammatory cytokines and the anti-inflammatory cytokines. Remember that cytokines, especially interleukins, they play a major role in this along with the TNF alpha. Now, there are few interleukins which are pro-inflammatory. That means they trigger the inflammation locally because inflammation is necessary to stay alive. And the inflammatory interleukins are interleukin 12, interleukin 6, interleukin 2, interleukin 17, and interleukin 18. And the predominantly anti-inflammatory interleukin is interleukin 10, which has got a job of not only curbing down the inflammation locally, but also it has got a job to go back to the site of the production of these defense cells and inhibit those colony-site stimulating factors to reduce down the production of macrophages, lymphocytes, neutrophils, basophils, eosinophils. But what happens is this, that in a severe COVID time, there is a dysregulated secretion of pro-inflammatory cytokines, and there is a dysregulated or excessive production of the anti-inflammatory cytokines. And as effect, there is a dysregulated inflammatory response, which is also popularly called as cytokine storm. Actually, cytokine storm is not an actual correct terminology. The correct terminology should be ideally the dysregulated inflammatory response. And so this synchronized symphony, which is on for our local protection, starts becoming dysynchronized. And it leads to ultimately a blizzard of outrage immune response, which is triggered in the pneumatocytes or the type 2 pneumatocytes in the lungs, and then spreads systemically, leading to what we know as a systemic inflammatory response. And this happens basically because of the pathological immune response, what we know as immune paralysis, because of an excessive production of the antibodies, which are probably useless. There is an excessive production of the killer T lymphocytes, which have a sort of an autoimmune destruction. Also, there are abnormal responses from the granulocytes, monocytes. There is an excessive stimulation of TNF alpha, which is a very potent pro-inflammatory cytokine. Along with that, there is a huge amount of lymphatic dysfunction setting, which ultimately leads to the dysregulated immune response all over, and which leads to a cytokine storm. So this storm, which is generated because of this dysregulated immune response, can be weathered, can be curbed by what we know as immune modulator or immune dampeners. And that was the concept where monoclonal antibodies were thought to be useful, especially managing this cytokine storm or dysregulated immune response in the COVID-19 times. The concept is not new. The concept is pretty old. We have used this monoclonal antibodies, while managing some of the autoimmune diseases like rheumatoid arthritis or SLEs, or even the previous viral infections like MERS and SARS. That time also we have used them. So this cytokine inhibitor biologics, or what we call it as monoclonal antibodies, are the biologics which have come into the limelight and which are the need of the hour. So let's understand what are these therapeutic antibodies, what we popularly know as monoclonal antibodies. They are the need of the hour. They are needed to treat most of the critical illnesses. And they offer a promise of what? The promise of target specific therapy. Gone are those days of a broad spectrum therapy. So you no more have to use atom bombs to destroy your enemy. You require a precision target specific therapy. You have got that promise from the monoclonal antibodies that we have. They are usually either human or they are engineered by genetic engineering or various engineering processes. And they are armed to deliver the target specific therapeutic response at the precise target. Mind well, most of the monoclonal antibodies are IgG type of them antibodies which are used for the production of the monoclonal antibodies. So let's have a overlook of what are antibodies. The antibodies are the antigen binding proteins. Now, what are these antigen binding proteins? These are the proteins which are secreted primarily by the B cells or the B lymphocytes. So whenever an antigen enters the blood circulation, and as it is sensed as non-self or foreign, the B cells they go and sense it and they trigger the formation of plasma cells. And these plasma cells, after conversion from B cells, once they form a sort of a body defense response in the form of antibodies. And these antibodies they circulate in the blood where they serve as an effector of the humoral immunity by searching and neutralizing the antigen and marking them for elimination. Remember that antibodies do not kill. What they do is they mark the antigens which are foreign or non-self and they present or sometimes help to present these antigens to the killer system or the killing mechanisms of the body, like the natural killer cells or CD8 cells, which actually have got the destructive properties. They work in tandem with antigen presenting cells. What they do is this that they encode or go and just engulf the antigen inside them by a process which is called as opsonization. Now this opsonization means in simpler words it is like making it edible by a sort of what we call it as a chocolate coating. So they make these antigens edible by the natural killer cells or the CD8 destructive cells. And this is what, or the cytotoxic T cells we all know. And this is what is the job of antibodies. We all know that there are 5 different types of antibodies which are naturally produced in the human body. Out of them, 3 are monomeric. That means they only have one structure. Those are IgG, IgD, and Ig. Whereas IgA is dimeric, it has got 2 arm structures or the 2 different molecules attached with a J junction or the J chain. And the biggest one is the IgM, which is pentameric. Now, why it is important to have pentameric? Because these all different arms of this antibody IgM, they are equipped to sense the different type of antigens. Now, these antigens, if you really look at the schematic diagrams of antibodies, you will realize that the antigens are sensed by this Y arm of the antibody, which actually cling on or hinge to the antigen sensing structure on the antigen. They are called as epitopes. And where there is a constant fragment, what is called as an FC fragment, that is the fragment which is constant, which actually is attached to the cell of immunity. So what these antibodies they do is this: they go and sense this antigen sensing area, what are called as epitopes on an antigen, and they go and attach and immobilize or neutralize that antigen. They fix it. And these epitopes are different types. You can see on this diagram that look at the different shapes and sizes of epitopes. And depending on these different epitopes, different types of antibodies are produced. So these are called as, so for each one specific epitope, one specific type of antibody is produced, and these are called as monoclonal antibodies. So antibodies which are produced for one specific type of epitope of antigen, they are called as monoclonal antibodies. So B lymphocytes have an ability to produce antibodies against either single antigen or to the multiple antigens. So when it produces the antibodies against multiple antigen, usually what is the classic example is IgM acute response. They are derived from multiple B cell lines and they are called as polyclonal. They are broad spectrum, they are not target specific. And they are monoclonal antibodies which are derived from the specific single B cell line. And what is most important is They are very, very specific. When an antigen triggers the B cell, it gets converted into plasma cell, and plasma cell memorizes that antigen and keeps on producing antibody against that specific antigen. This is called as the monoclonal antibodies. Now, monoclonal versus polyclonal, how do you differentiate? Monoclonal antibodies are derived from single B cell line. Polyclonal antibodies are derived from different B cell lines. Monoclonal antibodies are reproducible. They are predictable in its action, and they are potentially inexhaustible because you can keep on producing again and again by the genetic engineering. Where polyclonal antibodies, they are difficult to produce, they are not predictable, and there is a huge batch to batch variation which can happen. So which makes antibody response sometimes less effective when you use polyclonal antibodies. And monoclonal antibodies are very easy to develop outside, and especially they are beautifully used in the immunoassays for the various investigations. And this technology is used to produce monoclonal antibodies is called as hybridoma technology. I'm not going into details about it, but the concept is this, that you know the mouse is injected, the spleen of the mouse is injected with what we call it as an antigen, and this antigen produces an antibody against that a particular antigen in the spleen. So these splenic cells are extracted from the mouse and then they are fused with myeloma cells. Now, why myeloma cells? Because myeloma cells have got a long life and they keep on producing antibodies regularly and they do not die. But unfortunately, the myeloma cells which produce those antibodies sometimes may not be very effective. So that is the reason why the antigen specific antibodies, which once they fuse with the myeloma cells and they are grown into what we call it as a hat culture medium, they produce multiple monoclonal antibodies for the different types of epitopes. So the epitope of your interest, the antibodies for particular epitope of your interest are specifically picked up by a technology which is called as an expanded positive cloning, and then they are actually extracted and used for the therapeutic purpose. This, I know that this is a little complicated. I'll talk to you in details whenever I get a specific talk on the hybridoma technique. It's a very interesting technique. And for that, in 1974, George Kohler and Cesar Milstein won Nobel Prize on that. So biological products which can be obtained, or these monoclonal antibodies can be obtained from various different mechanisms, or they can be synthesized by recombinant DNA technology from either viruses, fungi, bacteria, toxins, antitoxins, vaccines, therapeutic sera, blood components, or even sometimes from the natural proteins or allergen products. What they do is they pick up the IgG structure outside and they do what we call it as a genetic engineering. They change these different light chains and heavy chain parts and keeping the constant fragment specifically constant. And they generate and get these antibodies. Now, if the antibodies are isolated purely from the murine specific situation, they are called as murine. Chimeric, that means that it is 65% like human and the rest of the 35% is like murine or mouse originated. Humanized means more than 90% is like human antibodies, the structure is like human antibodies. And fully human, that is 100%, is that means that they completely are identical with the human antibodies produced. Now, why it is important to understand these different very species specific differentiation? Because depending on the species of isolation, the immunogenicity depends of the response to these antibodies they vary. Urine antibodies have got higher immunogenicity, whereas humanized or complete fully human antibodies have got much lower immunogenicity. Then, how do you identify which antibody is from which species and why it is used? So there is a typical what is called as a Boron's guide for the monoclonal antibody nomenclature. Now, what is this monoclonal antibody nomenclature? It is the naming system. For the first time when I heard the term itolizumab, I was surprised. Why are they running out of the names or something like that? Then when I looked deep into this, then I realized that they have got a specific way of labeling these antibodies. Like there is a prefix word, then the middle interfix, it talks about the target where these antibodies are going to act. Then another interfix will talk about the source, and the last one will be suffix, and this suffix is usually MAB. Whenever there is this suffix of MAB used, that means that it tells you that these are monoclonal antibodies. Say like example, there is a prefix, and if the antibody target is bone, then usually the interfix would be either O or OS. Similarly, if it is immune modulating, then the interfix would be L or IL or LI. And that is the reason why if you have heard that name, tocilizumab or itolizumab, that means that those are immune modulating antibodies. And then whenever there is an interfix which is using U, that means that it is completely humanized. If it has ZU, that means it is humanized, that means more than 90%. If it has got XI, then it is chimeric. And if it has got A, that means it is murine. Now I'm going to give you examples of this. Okay, and just see here. Infliximab, XI. Inflict LI, that means it is immune modulating. XI, that means it is chimeric. Infliximab, bevacizumab. So bevaci, CI, circulation. ZU, humanized. So bevacizumab. Rituximab, RITU. TU uses for tumor. XI means chimeric. And this is the way the whole antibody nomenclature happens. So the examples of different murine, chimeric, and humanized antibodies are like, murine, chimeric are infliximab, apiximab, rituximab you are used. Humanized are, we are using this in COVID times, are tocilizumab, itolizumab, or trastuzumab. And completely human are adalimumab, especially in the patients of acute autoimmune disease. If you look at the role of antibodies in the immune system, there are various targets these antibodies they target to treat these autoimmune diseases. And especially looking at COVID, we have antibodies which actually target interleukin 6, interleukin 1, TNF alpha, to even the systems like monocytes, or even the target on T cells can be addressed by these different antibodies. The monoclonal antibodies are also available for another reasons, like for RAS inhibition, for immune modulation, basically for production of IVIG, or against the granulocyte monocyte colony stimulating factors, or for the anti-rheumatic factors, or for interferons, or even for statins. And that is the place where you can see here on the graph that the antibodies like canakinumab or anakinra, all of you have heard about anakinra, and it's used in rheumatoid arthritis, or tocilizumab, or sarilizumab, or cetuximab. These are all the monoclonal antibodies which have been tried and used. What is the best time to use them? Lastly, is that ideal time to use these monoclonal antibodies is the time when the viral response is slowly fading away and a post-inflammatory response phase starting. This is the ideal time to use the monoclonal antibodies. What are the monoclonal antibodies that we commonly use in the patients of COVID? Are interleukin 6 inhibitors or CD6, that means that T cell inhibitors, broad spectrum, or interleukin 2 inhibitors, or sometimes the JAK inhibitors. Tocilizumab is one such specific interleukin 6 receptor blocker. Now, why it has come into the picture? Because it blocks the interleukin 6 receptors, thereby curbing down this pro-inflammatory response, which is generated and which is blamed for causation of the cytokine storm. And its role has been proved pivotal. If used at the right time, they really serve as the life-protecting drugs. Similarly, I also have used a lot of itolizumab in my practice, especially when tocilizumab was not so freely available. I chose to use itolizumab because it is a broad spectrum T cell inhibitor, and it inhibits what we call it as a CD6 protein, which is actually the cluster determining protein, which actually tells CD6 is the cluster determining protein, which tells that if a CD6 is present on the cell, that means that it is a T lymphocyte. And this inhibits that naive T lymphocyte, so it has a broad spectrum blockage of T cell response. And we all know that T response is very, very important for this acute immune response. So if you differentiate between the CD6 inhibitors and interleukin 6 inhibitors, you will realize that itolizumab is much more broader spectrum monoclonal antibodies, which causes downregulation of interleukin 6, interleukin 2, interleukin 17 or TNF alpha, whereas tocilizumab is more specific. That is the reason why tocilizumab is more broad spectrum and long acting. Tocilizumab is very, very target specific and short acting. Why it is important to know? Because if you give itolizumab, the possibilities of secondary infection, fungal infections, reactivation of tuberculosis is much higher in comparison to the use of tocilizumab. There are few limitations of biologics that you are supposed to know, that they are very expensive. If they are used at the right time, then only they are beneficial. But if you use them at the wrong time in the wrong dosages, then probably they can trigger the secondary bacterial infection or even the fungal infection. They work as a severe immunosuppressant drugs. So when they are used with corticosteroids, they can further worsen the chances of development of hospital-acquired infections. Also, there could be an activation of the tuberculosis, which is a genuine potential risk. And they do have what sometimes the hepatic or endocrinal or hematological or sometimes the cardiovascular side. So to conclude, remember that COVID-19, as we know, is more recognized now as a syndrome of not only the viral replication, but the dysregulated host immune response. Effective pharmacological immunomodulation strategies may definitely will help in alleviating the syndrome progression, especially shifting the patients to the cytokine storm. But remember that these are all very, very sharp-edged weapons. These are all double-edged swords. You need to use your smart clinical acumen to use them correctly. The huge future of this genetic engineered monoclonal antibodies. I'll be definitely happy to tell you more about this when I get a chance. But this is a gross overview where you can, usually use this. What is the future? There is a carbohydrate segment which is there, or what is called as an alcohol based structure which is there at the constant fragment, and to this carbohydrate molecule you can attach even the isotopes, you can attach even few drugs or even the genes which can go specifically. So there is no more broad radiotherapy needed. You just attach a radioisotope there and it will go and deliver the radiotherapy to the target tumor site. You can attach a specific DNA there and can make that DNA to really change the signaling at the local site. There could be a cross-linking of two MABs, which will help you in giving the action on two different targets at the same time. It can be used in multiple areas. And I can tell you that they are going to be the future. They are the future for radioimmunotherapy. They are the future for the angiogenesis inhibition. They are the future for the tumor-specific IgG therapy. They are the future for CAR T cell therapy. They are the future for the biospecific antibody therapy. I end my talk with this fun fact that immunology per se till today have won 19 Nobel Prizes, highest in any class of the medicine or physiology. If you really weigh the total immune cells and the proteins we have in our body, they will be around 1.2 to 1.5. So the memory cells can live up to 30 years. So remember that the memory can stay as long as 30 years. And the most mesmerizing fact, our human immune system is able to sense approximately 1,000 trillion immunogens and keep them in memory. Thank you very much once again for the patient.